Good morning and namaskar from India. So I stand here to talk about three stories interconnected, one slightly bigger, one smaller than that, and one just a teaser as a concept going forward. So I come from Institute of Genomics and Integrative Biology, situated in Delhi, and this is the Genomics Institute in India. So what do we work on? So as an institute, we work on certain 6 focus areas, which includes infectious diseases, systems biology, big data, chemical biology, and we have graduated to a lot of milestones in terms of genomics. What do we work on as a lab? So we work on a concept of integrative genomics of host-pathogen, and we hope that the research we do in the lab will provide some hope to the public health. And we have 5, 6 pillars on it, and we work exclusively using clinical samples with a lot of clinical data to understand why same or similar pathogen infection leads to dissimilar disease severity, like mild, moderate, severe, and in unfortunate cases of mortality as well. So for that, we use an integrative genomics approach, wherein we look at the viral genomes, we look at host response in terms of protein coding, long non-coding RNAs. We look at splice isoforms. We look at multi-omics of single cell. We look at co-infections in terms of when a primary infection happens, we also have resident microbes within us. So how does dynamic works vis-a-vis the primary infection and the co-infections? And then we put all of this together to understand that whether we understand why differential disease severity happens. So what I'm going to talk today is about 2 interesting stories, which one has completed its journey in terms of published in iScience, and another is an ongoing process with iScience before they accept it. And what do they talk about? So one of the story talks about that traditionally we know that dengue virus produces one polyprotein which gets cleaved into different genes. But based on genomics evidence, we think that there are possibilities of alternate ORFs in dengue, which gets activated under different situations, and that possibly is one of the reasons that why we have different disease severity vis-a-vis dengue infections. So for undertaking this study, we did RNA-seq for the host total RNA, and we looked at the reads mapping to the dengue virus, and we did some normal stats in terms of ATGC differences between the different dengue serotypes, and then we found that there are certain regions of the dengue genome which are a bit different from the other regions of it. But then the primary question which we wanted to ask is that whether there are regions in the dengue genome which may contribute or which may be more used as alternative ORFs in different individuals, and whether they can be reasons for differential disease severity. And we did find some evidences around certain regions of the dengue genome. But that was more like as an appetizer for us to look into the actual question which we were looking. And what was interesting for us was that when we looked at the mutations in the dengue genome and looked at the potential of that, whether those mutations are capable of producing different codons for dengue in different individuals, and we found that yes, indeed, there are preferred codons in dengue compared to the human host in which they were present. And when we thought of taking it forward in terms of what does it translate into, probably the most important figure is this one, where you find, if you see the middle green one, you see that it's a dengue genome, where reads of the dengue genome have different peaks and troughs. And we wanted to understand that why these peaks and troughs are. And for that, what we did was we tried to look into that the mutations which we identified in the dengue genome, whether they are giving rise to potential alternate ORFs, and whether they have the potential to translate itself. So then we'll match this RNA-seq data with the Ribo-seq data as well, and we did find evidence of some of these peaks actually overlapping with the Ribo-seq reads as well, so which essentially means that these ORFs have a potential to be translated in the situation where they may be important for the pathogen to survive in different microenvironments, and that may underlie differential disease severity. And these are, when we zoomed into specific regions, you can see that there is much more RNA-seq reads for those specific regions, and they overlap with the alternate ORFs identified through Ribo-seq. So when we summarize all of this in terms of why would a dengue virus make alternate ORFs, and we found that many of these alternate ORFs have the potential to make pore proteins, which may be useful for the virus for its survival within the host. So that's a story which has seen the light of the day. And when we summarize all of these findings, it takes us a step forward to see that why there are still challenges in terms of dengue vaccines, and how understanding an unexplored part of the dengue genome may help us to reach closer to the idea of having vaccines for dengue, especially for endemic countries like India, where there are extremely high cases of dengue. So coming to the two small stories before I say thank you, one concept which we are also taking forward is, can we, as all of us will agree, that genomics is a skill set which requires funding. Can we also work towards economics of genomics? And throughout this day and a half which I have been here, I have been hearing more often about 30x human genome vis-a-vis having functional understanding. So we took that question in mind and said, well, do we require 30x human genome coverage using nanopore sequencing as well? Because 30x genome coverage is something which we have traditionally understood with short read lengths of 150 base pair paired end. But if we use nanopore for its long read potential, where in our lab, for example, we have found median read length of 10.5 kb, and there are much more longer reads than that. Should we revisit this idea that do we need 30x coverage, or we are good with 15x, 10x coverage using nanopore long reads as well? So we did invest a bit of time in that regard, and we did work on the optimization, both on the experimental and the bioinformatics part of it. And we did achieve really good results in terms of making functional understanding vis-a-vis 15x coverage of the human genome. And now we are taking this work forward in terms of sequencing the human host vis-a-vis the dengue infected individuals. And we did find a very uniform coverage across different chromosomes of the human genome. We didn't miss any part of the genome because we are covering in that front. And then we also did differential coverage and looked for whether that impacts the SNP calling or structural variants. And we found that 15x coverage is good enough given the read length we have. So that's that's a concept which we think we will be extending further with more studies, that how do we take advantage of the longer read lengths of nanopore as well. So the last part of the work, which is a work in progress, wherein we asked a question that given that we have longer methodologies, can we ask a question that when we do RNA-seq, different genes are covered differently in terms of the gene length. So a gene is, say, 1 kb, or a gene is, say, 250 base pairs. Based on how much of the gene length is covered in terms of the RNA-seq, does it indicate in terms of the importance of that gene vis-a-vis the disease, and whether that has an impact on the differential disease severity? So we worked on this both experimentally and bioinformatically to come up with an understanding that based on how much of the gene length is covered during any particular disease condition, that has an impact on the disease severity as well. And this is a work which is, we are loving interaction with the editor and the reviewers in terms of why we think we have a reason to believe that way. And we have done a large number of hospital-admitted patients RNA-seq to be confident about what we are speaking. So these are evidences that we find that in certain cases, we see that in the severe patients, the genes which are shorter in length are covered more, and these genes have a specific functions to do. So that's a work in progress. We're happy to share when it comes back. So before I say thank you, no work is complete without the anonymous patients who participated in this study to give the samples, the students who did all the work, and the funders who helped us to do all the genomics activity which we do in the lab. So thank you for your patient hearing.